Okay, well, I want to go ahead and get the program started for tonight. Very pleased to see some friendly faces out there and welcome some new people who are joining us for the first time. My name is Felika Ahostin Bryant. I'm the director of the Native American Educational and Cultural Center at Purdue University. And we have two wonderful guests that are here going to be sharing their research. This particular presentation is called the SIGP webinar, Highlighting Indigenous Researchers. And this is a part of our Sloan Indigenous Graduate Partnership program. So to begin with, I have a short PowerPoint presentation, and then at that point, we're going to open it up and have our two presenters begin. So here we go and share. Okay, I don't have any, no sound there attached. So again, welcome to our evening. We're broadcasting across several time zones, and so if you're able to join us, I do see some people from the Mountain Time Zone. I did also advertise this so we could span across, like I said, all the way from Hawaii, all the way to the Eastern Time Zone. So thank you all for joining us. Excuse me. As we always do with our presentations, I always feel it's important that we offer a land acknowledgement. Before Purdue University was established, before the state of Indiana was established, the university was built upon the traditional homeland of the Potawatomi, the Lenape, the Miami, and the Shawnee people. And for me, coming from the Navajo Nation as an Indigenous woman, I really feel it's important that we acknowledge the original caretakers of the land who were here long before, who left so many footprints and who taught us so much about the land and the environment. And we do acknowledge also that there are additional tribes that we did not have on here, but again, due to forced relocation and colonization, many of these tribes have dwindled down. So there were some additional tribes that were in this area, but for the most part, we want to recognize these. And I would also encourage you, for people that are here, if you wouldn't mind going to your chat and just adding the home where your homeland is at, so that way we can see where we're again broadcasting from all different parts of the country. With the Sloan Indigenous Graduate Partnership Program, this is funding received from the Alfred P. Sloan Foundation, where we offer financial and academic support to Native Hawaiians, Alaska Natives, and Native Hawaiian students who are seeking a master's or a PhD in a STEM discipline. And we've got some information here about the program benefits. Funding is over the course of the student's degree program. We encourage our students to focus their research on a topic that's relevant to their tribal community or to a tribal community. So you'll hear some presentations along that lines. And then we also provide professional development opportunities. Many times we'll encourage students to attend and present at national conferences, and we'll provide some financial support as well. And we also provide mentoring for all our students. Program eligibility, students have to be connected to a tribal organization, Native American, Alaska Native, or Native Hawaiian, pursuing a master's PhD. And for the Purdue program in particular, students have to meet the admission standards in order to get into the university. And at that point, we then work with them with their Sloan application. The Sloan program is part of a national program across the country. We are very pleased to work with some wonderful partner institutions, including University of Alaska, both their Anchorage and Fairbanks campus, University of Arizona, University of Montana, Montana State, Montana Tech, State University of New York and the Environmental and Forestry program, as well as the University of North Carolina, Asheville campus. Our program highlights specifically for the Purdue program. We currently have 13 students in our program. At our largest capacity, we're usually at 18 to 20, but we had a large cohort of students that graduated this past year, 2020. Since we've had the program at Purdue, we have produced 45 scholars, and we have an impressive retention rate of 93%. Again, this particular program is open to students across all these different campuses. And I'm going to spend a little bit of time here because I really want to make sure we give a shout out to all our partner institutions across the country. If you're interested in learning more about these institutions, I am providing some information, email addresses, as well as contact people. If you'd like more information, just feel free to kind of pop a note in the chat, and we can follow up with you. But we do really appreciate all the work that all our partner institutions do across the country. So with that, I'm going to end this presentation, and I'm going to begin by sharing our first speaker for tonight's event. We are pleased to have Makani Fisher, who is a master's student at Purdue University, and he is studying entomology. Makani, I always have interesting conversations. What I love is the fact that he carries around plastic bags in his backpack all the time, so that way he can come and show us all the wonderful bugs that he's acquired along his journeys throughout campus. And yeah, I really appreciate Makani's he mentioned that I asked what got you prompted to go into entomology. He says, I never grew up because I like bugs. So I think you see that same energy and enthusiasm carrying into his master's program. So Makani, I'm going to go ahead and turn it over to you, and you can begin your presentation.
All right, can everyone see this and hear me? Are we good? Okay. Yeah, so like Felica said, I really enjoy moths and insects, so I'm gonna start my timer here, just to make sure I don't talk for too long, because I get really excited about all of this. Just one quick correction is that I'm a PhD student here, but I will be talking about things from my master's as well as the PhD work that I do here. As you can see here, the presentation of the title of my presentation is The Evolutionary Ecology of Lichen-feeding Tiger Moths. And there's been a lot of people that have been involved in these different parts, and I thank them a lot for that. Let's see if I can advance here. All right, so also like Felica said, that's my answer for every time I get asked, why I went into entomology, and it's really because I never grew up. And I've loved bugs and insects ever since I was little. It all started with a monarch caterpillar when I was about 8 or 9 that I brought home. And then we were actually able to take it to my elementary school class, and we reared it or grew it into the butterfly. That's what really got me hooked on these fascinating insects and the system. But it was really a hobby and I guess an interest more until probably the second half of my undergrad, when I met this man, Riley Nelson, who showed me that it could actually be a career path, and that it still amazes me that I can, I say play around with insects. I should say study insects for a living, but we have a lot of fun. But yeah, so this Riley Nelson was my undergrad advisor as well as my master's advisor. And as you can probably guess from seeing him in the river, we started with aquatic insects. So this is just kind of one of the weird aquatic insects that we investigated. But I always had this drawback to wanting to go back to moths, which are Lepidoptera. That's the grouping of moths and butterflies. And in particular, tiger moths. You can see here an array of tiger moths that they have a lot of bright oranges, reds, cool colors, patterns, and they're just really fascinating. So that's what I got into in my master's thesis, is started studying these tiger moths. And they also have a lot of cool behaviors. Here is some behavioral stuff done by Dr. Dowdy, who's a postdoc in my lab, that he investigates heavily the acoustic behaviors of these moths. A lot of the tiger moths are not only able to hear bats and their acoustic sonar, but actually able to reply to them. They can either tell them that they don't taste good because they have a lot of chemicals, or in some cases, these moths are even able to jam bat sonar, so they hide from the bats. So they have a lot of cool behaviors, and so that drew me even further into studying these awesome moths. The group of tiger moths that I study specifically are the lichen-feeding tiger moths, or Lithocyane, that's how you say that there. That's a group of tiger moths that feed on lichen. There's about 3,000 species worldwide. And I just want to point out, I guess, that it's the caterpillars. Hopefully get this photo to load. There we go. It's the caterpillars that specialize and feed on lichens, mosses, liverworts, and algae. As you can see here, these are just some of the caterpillars I've had in the lab eating lichen. And just a quick recap on what lichen is, if you don't know. It's when a fungus and an algae come together and live in a symbiotic or a mutual kind of relationship. And they make a lot of different structures. You can see a lot of them here. I don't know if you can see my mouse, but there's a lot of them here. with different colors, shapes, sizes, all sorts of different variety. If you didn't know what lichen is before this, you've probably even seen it just walking outside on trees, rocks, things like that. And if you cut into one of them, this is what it looks like. It has the fungus on the outside and basically sandwiching in the algae in the middle there. So there are these kind of interesting, weird organisms, as well as another thing that makes them interesting is they have a lot of secondary chemistry. And these chemicals help protect them against things like solar radiation, but especially against things like herbivory from things eating them. There's a lot of chemistry and arrows going on over here, but the main thing that I want to point out from this, or the main takeaway from this, is that there is just a lot of chemistry, as well as that there's three main pathways that these lichens make this chemistry. And interestingly, despite this chemical defense, there are still a lot of invertebrates that feed on lichen. Let's see if that works. There are at least 12 different insect orders that do, as well as you can see here is a snail, and this is a millipede up in the top left. So there's still a lot of invertebrates that are feeding on this despite its chemical defense. But despite this broad radiation of feeding that happens on lichens, we still don't know a lot about these interactions, their evolutionary history, or even their ecological implications. So there's still a lot of unknowns with it. And that's what we look to fill with these lichen-feeding tiger moths, and that's why we study them. Tiger moth, or I guess lichen-feeding tiger moths are very interesting because they, like other invertebrates, are not only able to pass these secondary defenses, but they're also able to incorporate or sequester in chemicals from the lichen into their own bodies. And I think that as far as I know, they're the only invertebrates that we know of that do that. And so that's what our research kind of is geared towards. I look at the evolutionary history of this and what they might be doing with these chemicals and the ecological implications of this. The first part of my presentation, I guess, on my research here will be on host preference and how secondary chemistry might influence which lichens these caterpillars are eating. And then I will also discuss a little bit about broader implications that this lichen feeding could have in ecosystems and bigger systems. Oh, excuse me, spilled some water. But yeah, so I guess a little bit of history that lichen feeding is really cool, but it hasn't been investigated very much, especially not even within tiger moths. But there has been some work. So this is a little bit of research that was done by a group in 2003 that looked at some of this host preference stuff, where they used this genus or this group of moths called Elema. And what they did is they presented these lichens to them that had different chemicals, and they saw what their preferences were, which ones they went to, what they ate, and so forth. And what was interesting was that they preferred lichens that didn't have a lot of chemistry. They didn't really like the ones that had chemistry, which is really kind of interesting when we look at that we have found lichen chemistry in at least 40 different species of tiger moths. And so the question is, it starts to become, where do they get these chemicals then if they're not getting them from the lichen? We believe that they are. And so we looked at this in an evolutionary framework. This is a paper that came out, or a study that came out from a person in my lab that before I got here, but 2018, they looked at the evolution of these lichen chemicals within these tiger moths. This is a phylogeny. If you've never seen one of these, what it is basically a clustering or an organization of different species based on DNA data and evolutionary models. But it's a nice way to visualize and see what is related to what and how the evolutionary relationships play out. But beyond that, what these are useful for is to start to see patterns in different characters or behaviors. And we do that by looking at these characters by lineage. That's what each of these on the right-hand side, the Lithocena, Neuterina are, and stuff like that. And what these different pie circles represent are these different chemical groups that these insects have a preference for. And so what they found was that different lineages or different groups of caterpillars prefer different chemicals. And so that might explain why these past researchers who didn't put it in an evolutionary context might have missed that. It's because they might have given them the wrong lichens with the wrong chemistry. So we wanted to test, well, if we gave them something that they developed to evolutionarily prefer, would they then prefer that lichen? And so we set out to test that. This is largely what my master's thesis was, involving this moth, Cisthene angelus, in the southern deserts of Utah, beautiful red rock country. And what we did was we put out these lights, we attracted the moth, there's the little moth there, and we got eggs from it. This is just showing here the life cycle, the eggs to caterpillar to pupa to moth, but we grow them on lichen. And then here's the chemistry again. Don't get too bogged down by the chemistry. But what this is really pointing out is the lichen on the bottom right, A here, is the one that we hypothesized that they should like or prefer. It's the one that has the chemistry that this lineage or group of caterpillars was shown to sequester in that evolutionary phylogenetic study. And then we had some others that didn't, had some chemistry, but they were from different pathways. So we presented those to our moths in these little arenas where we dropped them down here. We watched where they went over a 30-day intervals. And what we found was actually that this red peak here is that they did have this, this preference for this lichen that we thought they would. But what was interesting is that after 10 days, they lost this preference and started eating other lichens like the bee lichen. And so we were wondering why this could be the case, and we looked at nutrition. Maybe these caterpillars have to balance getting the chemistry that they like along with the nutrients that they need to grow. And this, we found some data that confirms this. We took another batch of caterpillars and we grew some on solely one type of lichen, A, B, or C. And then after 45 days, we measured their head capsule to see how much they grew. And you can see that those that fed solely on the lichen that has the secondary chemistry that they prefer, they might prefer that secondary chemistry, but they grew to a smaller size. So they do have to balance that, getting the nutrition that they need, as well as getting that secondary chemistry that they prefer. And that makes context, taking it back to the bigger picture, looking at the environment that they live in. You can see a lot of different lichens, the gray ones, the green ones, the yellow ones, even these little black ones here. There's a lot of different lichens that are available to these caterpillars. So if they're able to utilize at least a little bit of variety, they're probably doing a lot better. So that was my master's thesis, and we kind of use now similar approaches on things that we do in my dissertation. I work on a different group of moths now, but they're in that same lineage. And we use that similar co-play of phylogenetics and evolutionary history and that framework to inform us about our ecological data and vice versa, to help kind of get a bigger picture of what's really going on with these lichen insect or lichen invertebrate interactions. What I do now in my dissertation is I work on a species complex in the genus Hypoprepia. If you don't know what a species complex is, it just means that there's some species that are kind of hard to tell apart from each other. And it's a little questionable on how many species there are. It can be like that at times. And you can see here, these are the two species that are in the complex, Fucosa and Miniata. And just looking at these images, it would seem pretty simple to be able to tell these apart. I mean, Fucosa has this yellow orangish red kind of hue to it, and then Miniata has this nice deep, rich crimson red to it. But then the issue becomes that there's all this variation in these different, what we call phenotypes, or their external colors and wing patterns. And you can kind of see that it can be kind of a little bit hard to tell which one's which, especially when these things start to co-occur in the same area. And you also have to keep in mind that these are the representative photos of these insects that show these characteristics, that a lot of times it's even more muddy than this. So it can be difficult to tell these apart. So one way that we do that, we did do phylogenetic stuff, and I will talk about that a little bit later. We did do it with what's called CO1, which is a little fragment of DNA, just one little gene, essentially. But the results weren't very helpful, or very conclusive on figuring out what is what. But we did also use that little segment of DNA in what's called a haplotype, which is a similar thing to phylogenetics, but it's doesn't use a whole lot of evolutionary models. It just clusters things based off that gene of how similar things are. So things that are identical will be clustered all together. So that's what these larger circles are. They just have more individuals that are all identical. And then the lines in between these clusters are the differences. And these hash marks note how many differences are between these different haplotypes. And this gave us a little bit more structure in trying to figure out what these different insects were, or these different species were. This is the Fucosa, this is the Miniata, but then all up into this region where they have the smaller circles, it's just all mixed still. There's not a lot of structure, so it pointed us a little bit in the right direction, but it's still a little bit of a mess. So another thing that we tried is dissections, looking at the internal morphology of these insects. We've done 52 dissections where we cut off and look inside the abdomen of these little insects. And we look at the genitalia. And the genitalia, because a lot of times those are conserved features, features that you can really hone in on to tell between different species. And they're sclerotized, so they're easy to pull out of the inside of the insect. But you can see how kind of crazy and weird looking these are. These are the male's genitalia on the left and middle, and that's the female genitalia on the right. And you can probably guess and imagine how difficult it is to sit down and try and figure out what are the characteristics on these genitalia. So that's still a bit of a work in progress, but a rough glance over this has shown that there's a little bit of structure there, that there might be those two species, Fucosa and Miniata, and maybe one other, but it's still kind of a bit of a mixed mess. So then we looked at geography. Really kind of what we're doing is we're just throwing a bunch of lines of evidence at this and trying to figure out where the story is, and where we can start drawing boundaries on where these species, what makes each species unique. And so the important things in here are that the stars, this is a map from, I guess I should say, a map from one of the older students in the lab. But the stars here are that Fucosa, that one species, and the triangles are Miniata. And so you can really see that they're just really intermingled in these areas. But these variants, or these subspecies, these weirdo kind of phenotypes, are kind of more geographically distinct. You have this kind of gray one that occurs up here, and then these more bright ones that occur down lower. So that might give us a little bit of insight into maybe what's going on, especially if we can't parse out that these are different species, for example. It could be that environmental factors or something in their diet around there is impacting their color. And maybe going from a red to a yellow to an orange really doesn't take that much effort to do. So finally, I guess bringing it all to what evidence and stuff that we do have, we did do some more phylogenetics with more DNA. Like I said earlier, the CO1, or the phylogeny based off that one little gene, didn't really yield useful results. But then we tried it later with a different technique that gives us like 740 different genes. So a whole lot more genes, a whole lot more data to play with. And we got a lot more structure and organization. And so you can see here, this we were able to get Fucosa separate from the other species, Miniata. And then there may be this, what is E here, this yellow little thing down there. That might be a new species. So we're getting a little bit more structure and a little bit figuring out what these different species are. But there's still a whole lot more work to do. What this plus sign in this box on the right is all of the different phenotypes that still have yet to be added into this phylogeny. And that adding those in, I think, will really give us a good picture on really what's going on with this species complex, and really like how many species there are, how do we distinguish them from one another, and how can we use them, for example, in our ecological studies, which is one of the main reasons that we want to figure out why or what the differences are between these species and what really are the distinguishing characters, is because we then use them to understand interactions, for example, between moths and bats. This species, or this, I should say, species complex, is well known for their unpalatability, or they're distasteful to bats. And we want to explore that further, but we really need to parse out being able to diagnose or distinguish between different species, so we can get the clear and proper story between this. And as also you can probably imagine, COVID has thrown a bit of a wrench into playing with bats, but we're optimistic. We're writing a grant actually right now to further investigate into these interactions. And I guess just a quick last little blurb about this. Another interaction that we're wanting to look at is between mantid and moths, because we know, like I said, we know that these moths are unpalatable to bats. However, we don't know how they are to any other predators like praying mantises that they may encounter in the wild. And again, knowing which species is which is helpful in that endeavor as well. And so, yeah, for now, especially with COVID, my apartment will continue to look like this. Thankfully, it doesn't look like this right now. It's just during the summer months, all of these little cups with a bunch of different little caterpillars that we grow on lichens, to start to tease apart these different interactions. And with that, I would like to thank everyone that has contributed to this, and yeah.
Okay, thank you so much, Makani, for sharing your information, and it's always good to hear what you're studying. And I always, again, like the fact that you carry around plastic bags in your backpack in order to pick up bugs along the way. So if you have any questions, just pop them into the chat, and then we can get to those questions here at the conclusion of our next presentation. So I'm going to move on to our next presenter. I'm very pleased to have Nicole Benally join us. She is a Purdue alum, as well as a currently enrolled Ph.D. or doctoral student at the University of Montana. Nicole received her Bachelor's of Science from New Mexico State University and her Master's in Agronomy from Purdue. And she's from the Navajo Nation. She currently is a Ph.D. student in Forestry and Conservation and a Food, Energy, and Water Nexus graduate trainee. So I'm going to go ahead and turn it over to Nicole.
All right, Phylogeny, can you give me a thumbs up if you can see my presentation? Okay, and it's just the presentation, not the little Zoom stuff? Okay, perfect. All right, thanks for the introduction, Phylogeny. It's always nice to present. So for those that aren't familiar with me, my name is Nicole Benally. Like Phylogeny said, I just started my PhD program here at University of Montana in the Forestry and Conservation program. And so my presentation is going to be a little bit different than like a research. It's definitely a journey for me through my experience through academia. I'll go more detail about myself again, talk about my master's research at Purdue University, and I am in my second semester as a PhD student. So I'll talk about where I'm going, and it's involving tribal food sovereignty, and then acknowledging everybody along my journey. And then at the end, I'll have my contact information as well. So like Phylogeny said, I am Navajo. I am Cisthene, born for the Moths clan, and my grandparents are from the Bitterwater and the Red Running Into Water clans. I was born and raised in Ganado, Arizona, and on the map above you can see the green area, which is the Navajo Nation. It's roughly the size of West Virginia. My experience as a little res kid was only growing up with Navajo people around me, so I did not know that other tribes existed growing up, and I grew up in a low-income household and a single-parent household, so we didn't go to like the nearby towns and cities as often, so my life was pretty much growing up on the reservation. And Yeah, so growing up on the reservation, and I dreaded going to town to the grocery stores in order to get food. So that kind of was my early note of becoming a tribal food sovereignty, researcher or activist, if you would say. The bottom picture is just a picture of myself and my family. We were growing some corn in our family housing apartment in New Mexico in Las Cruces. So like Flicka said, she covered my bachelor's and my master's degree, but I'm also an alumni of the Navajo Preparatory School, which is a college preparatory high school for mainly Navajo students, but it's open to any other student to also attend. They're heavily focused on the Navajo culture and the language, but also preparing their students to get ready for college and applying for scholarships. When I was graduating from Navajo Preparatory School, I was awarded the Gates Millennium Scholarship with the amount of extracurricular activities I was doing as a high school student and getting good grades on top of that. So I was very honored to get that scholarship, and that really set the path for me to even start college right after high school. So at New Mexico State University, I got my degree in agriculture, majoring in soil science. And I didn't start off as an agriculture person or even in soil science. I actually started as an engineer and switched over to environmental engineering. Took my first soils class, fell in love with it, and that rest is history, I guess. And then I went to Purdue University for my master's degree in the agronomy department, where I continued to be a Gates Millennium Scholar and a Sloan Indigenous Graduate Program Scholar as well, which Felika had covered before our presentations. And the pictures to the right are just some fun family pictures of myself and my family as we were graduating. And then also some fun little fieldwork pictures and me presenting early on in my career. After graduating with my master's from Purdue, I worked as a tribal agriculture and 4-H extension agent for about a year. It was with New Mexico State University. I was serving the Eastern Navajo Nation in the McKinley County area. A lot of my work in the beginning was 4-H, and then I was putting my agricultural knowledge and my soil science knowledge when I was doing having booth spots during events and working with grassroots programs that were working on tribal food sovereignty efforts, getting to know them, getting to know their presenters, getting to know how they were doing their program to reach the Navajo community, which is a very rural community. So I got to build that community and that networking there. And that kind of led me into my PhD, which is, like I said, focusing on tribal food sovereignty, where I am a Gates Millennium Scholar and SIGP Scholar again, but also getting into the realm of integrating food, energy, water, and basically a lot of biophysical sciences and integrating that into social sciences here, because University of Montana isn't the land-grant institution of Montana. So I'm getting a lot of social science background and skill sets right now, and I think that'll help me as a native scholar to incorporate the biophysical sciences with the social sciences. All right, so just going into my master's research, this is going to be a SparkNote version. For the sake of time, I'm just going to talk about soil aggregation, and I'll talk about that more in detail later. So my title was Soil Aggregation and Soil Carbon Measurements to Assess Cover Crop Improvements to Soil Health in Indiana. So just a few key terms. So soil health is the continued capacity of the soil to function as a vital living ecosystem that sustains plants, animals, and humans. And just like us being healthy, soil has to be healthy. And the most visual example I can give you guys is if you had 2 cylinders filled with water and you had these little wire meshes to hold a soil sample, a healthy soil, when you put it in, which is indicated on your left, would keep its shape. Like some of the outside will break, but it would most likely keep its shape in that shape, and that's healthy. That's considered a healthy soil. And then on your right, an unhealthy soil, which is usually like low in organic matter, has been tilled a lot, or just doesn't have like living organisms living in it or on top, once you add it to the water, the cylinder of water, it'll break up immediately and disappear in a matter of minutes. NRCS also has 4 soil health principles that is very popular in the ag community. So maximizing and continuing to have living roots in your field, minimizing disturbance, maximizing your soil cover, and maximizing the biodiversity that you have on your field. As far as cover crops, so a lot of people aren't familiar with cover crops, and cover crops is a new term, I would say, and is being practiced more in the Midwest, in the Corn Belt region, and is getting more research across the nation now. But cover crops are plants planted in the late summer or fall around harvest and before spring planting of the following year's cash crop. So basically, farmers are planting a plant or a variety of plants after they harvest their money crop. The top picture, you can see just a very good visual of after harvesting corn. So with cover crops, you'll still have a plant growing, your field will be green. And on the right, without cover crops, your field will continue to be fallow and bare and prone to erosion and stuff. Another practice with cover crops is not just using one type of plant, but also incorporating a lot of different species. And that is just adding diversity on top of ground and below ground. It's important for like pollinators, diseases to have a diversity on top, so you don't have like the same pests impacting your cash crop, and you're just adding diversity there. And then depending on the crops you have, they have different root structures, and you're also having like a zoo. You're basically feeding worms and other microorganisms underneath the ground. So they're creating these channels where water can infiltrate faster, and that's where the plants can get the nutrients. So just a quick overview, healthy functioning soils are able to cycle nutrients, store carbon and nutrients in soil organic matter, provide good aeration and promote root growth, like I was mentioning before, reduce erosion and runoff, which is very important in Indiana because there's a lot of rain that happens, and a lot of the fields are still tilled, but there are some that are doing no-till. Improve water storage and plant water availability, and reducing disease and pest problems. And then incorporating a cover crop, the benefits of that is also to improve nutrient cycling, increasing your soil organic matter content, which is kind of like a glue for soil. And I'll talk about that again in a moment. Improve soil physical and biological properties, reduce runoff and erosion again, improve the availability of soil water, and breaking down those pest cycles. So for my master's research, we did look at physical, chemical, and biological indicators, but for this presentation, I'm just going to focus on the soil aggregate stability, which is just looking at the physical aspect. So the objective was to measure and evaluate the soil aggregate stability with cover crop use and tillage across Indiana. And our hypothesis was that using cover crops will have a greater aggregate stability than the no cover crop treatments. And soil aggregates are basically the little soil clods. So if you were to grab some soil or dirt and the naturally forming PEDs, those are the soil aggregates. There's a little diagram to your right. And the way we measure that is getting the average size, which will be called the mean weight diameter. Our study included 24 sites across Indiana, 3 Purdue agricultural centers, 2 soil, water, and conservation districts, 12 farmer sites, and then 7 neighboring sites were added in maybe the year I just started. And the conventional comparisons are just neighbors that did till their soil and didn't use cover crops at all. So the way we collected our samples is we used a golf cutter, which is the picture to your right. It was to get uniform samples throughout the huge fields. We collected 4 samples in each treatment plot at a depth of 5 centimeters. All of the samples were taken early summer, and between cash crop rows, we avoided where the actual corn and soybeans were growing, and we avoided the areas that were disturbed by animals or by people, our foot traffic, as well as like wheel and tractor traffic. So once we got our samples back to the field, we sieved our samples, and you can see it on the picture to your right, through an 8mm screen. We let them air dry. Once they were air dried, we sieved them again through a 2mm screen. For the bigger project, we kept both soil fractions, but for the soil aggregate stability, we just needed the 2mm to 8mm soil PEDs or soil aggregates. So again, the way we measured soil aggregate stability, I'll be referring to AgStab for the rest of the presentation. We only used the 2 to 8mm fraction, and I collected my samples 2016 and 2017. And again, we calculated the average size of the little PEDs, and they're called the mean weight diameter, and we used the wet sieve method. And here's a little video just showing the oscillator. So what we do is we have these 4 series of sieves that we tower and stack on top of each other. We add a portion of our soil sample to the top, and then the oscillator will keep moving up and down, sieving the soil sample in water. And then once a certain amount of time, we used 10 minutes, we took them out, and then we weighed each soil size fraction, and that's how we calculated our average size. Now looking at our treatments at the Purdue Agricultural Centers, the PACs, we had more, we had more, I guess, say on what we really wanted with the treatments, because there's multiple projects going on. So we had a corn and soybean rotation. Before corn, we had 3 different cover crop, Okay, sorry, the door opened and the kids were saying hi. So before corn, we had 3 cover crop treatments. The oat radish was one treatment. The oat radish, cereal rye, crimson clover was another treatment. And the last one was a no cover crop treatment. Now those treatments rotated with the neighboring treatment. So for example, before corn, the oat radish treatment would then be planted to cereal rye before soybean cash crop. The farmer sites, the farmers chose their cover crop, what cover crop they planted, but most of the farmers we have been working with had a no-till system with cover crops and a no-till system with no cover crops. And then when we added those conventional neighbors, they used some sort of tillage and had no cover crops growing on their sites prior. For our statistical analysis, we used ProcMix and SAS. We analyzed each site separately. We had 3 to 4 replications at the Purdue Agricultural Centers. The blocks were randomized, and our fixed variables were the year, cash crop, and the cover crop treatment depending on its cash crop. And then at the farmer sites, again, we had 2 to 4 replications where the blocks were randomized, and the fixed variables were year and cover crop. When comparing the farmer sites with their conventional neighbor, we just used a t-test. So just for time's sake, we're just looking at the cover crop treatments, the no cover crop treatments, and their neighbors at the Purdue Agricultural Centers, and then at the farmer sites listed below. So on the x-axis, you have the Purdue Agricultural site. So you have DTC, which was only sampled in 2016, NAECC, which was sampled in 2016 and 17, as well as CPAC in 2016 and 17. And then the cash crop that was currently growing when we were sampling, along with the cover crop treatment. On the bars, all the green bars are the cover crop treatments, and then the yellow bars are the no cover crop treatments. And on the x-axis, again, is the aggregate stability measured as a mean weight diameter in millimeters. So at DTC and NAECC, we didn't see any significance between our treatments, but we did see a significant difference at CPAC, and we were hypothesizing that DTC and NAECC were more north of northern Indiana, located in northern Indiana, where the seasons are shorter for the cover crops to grow, so we couldn't really detect those differences. But at CPAC was at the southern part of Indiana, where we had longer seasons for cover crops. And looking at the treatment specifically, the cover crop mix was significantly different than the no cover crop in the corn cash crop season. And then when the soybeans were growing, both the cover crops were significantly greater than the no cover crop treatment. So looking at this more in detail, just looking at what was growing at the time and what was growing before. Remember, I was saying that these are corn soybean rotation. So overall, when we sampled during soybeans, there was still corn residue left on the ground. So we think that played a bigger impact to, like, adding residue to the soil, increase the mean weight diameter or the aggregate stability than when sampling in the corn season. At the farmer sites, again, we have the farmer sites at the bottom and the years they were sampled, and it's simply a cover crop and no cover crop treatments. So at Brock-Smith and DeSutter, we saw more significant differences than at Shooter, Stahl, and Vilwalk. Brock-Smith and DeSutter had more years of using cover crops. So when you use cover crops, it is a long-term kind of commitment to see those results. And Brock-Smith and DeSutter had over, I think, 10 to 15 years of using cover crops. So we were thinking that's why we've seen those significant differences. As at Shooter, Stahl, and Vilwalk, they barely incorporated cover crops into their systems. So during my time of researching, we probably didn't detect those differences. We didn't detect those differences then, but later on we could if they kept using cover crops. Now comparing the farmer sites with their conventional neighbors, again, we use a t-test. So looking at the conventional neighbor and then the cover crop treatment, and then the conventional to the no cover crop treatment, we saw that there were significant differences between the conventional neighbor who was tilling their field and didn't use cover crops than the cover crop treatments. And then there was no significant difference with the no cover crop treatments and the conventional neighbors, except at Vilwalk. And Vilwalk was a little bit, that one was just a case-by-case, site-by-site experience. So I would have to look through back my thesis for that one. Now, like I said, my educational journey is I don't know, long-term, and having that experience at Purdue, working with farmers, learning how to soil sample and learning how to, plan, research treatments and how to write those really impacted my experience being an extension agent before becoming a PhD student. And then on top of that, learning, the missing links or the missing holes as an agent there is for support for tribal communities. So, which is why I wanted to focus on tribal food sovereignty for my PhD work. The definition for food sovereignty is the right of people to healthy and culturally appropriate food produced through ecological sound and sustainable methods, and their right to define their own food and agricultural system. And my perspective of food sovereignty, especially working with Indigenous people and through my coursework here as a PhD student, is that food sovereignty isn't a new concept. As Indigenous people, we had already established our own food systems where we originally were, like the Three Sisters, which is kind of like a new technique being recognized in agriculture and academia, but really those are practices that Indigenous people and many tribes in North America already practiced before. And also like dry farming, which is common in the Southwest, fishing for fish and hunting for wildlife and harvesting herbs and other roots and other medicinal plants on our rangelands. So my lens is looking at food sovereignty in the past, which I just briefly explained, looking at it presently, making it work and seeing what other grassroots foundations are doing today, and also looking at it as a research perspective, but also as an extension agent, like what resources do tribal communities have? And that also impacting the future. So in general, tribal food sovereignty challenges that I've come across in literature and from being like an Indigenous student, an Indigenous person, is genocide and relocation trauma that plays a huge impact on tribal communities when a lot of them were, it's just a lot of trauma. Some people or some tribes were relocated, like Felika had mentioned prior, where all their knowledge that their ancestors had was just pretty much wiped away because they moved to a whole new area. So they're relearning, or they have that gap in their knowledge. Also, fighting current mineral, land, and water rights is very challenging for a lot of tribes now. Looking at our treaties, looking at and recognizing where their tribe and their ancestors were prior to colonization. And a lot of literature incorporates tribal food sovereignty in food security and in food deserts. I grew up on the Navajo Nation, so we have, I think the last time I saw, we had like 11 to 12 grocery stores on the size of West Virginia. And there wasn't much agriculture all over the Navajo reservation, except in certain areas where you get to higher elevation or more north. So I would consider that a food desert. But being here at University of Montana and talking to other Native scholars that are from this area, they consider themselves a food desert too, just because grocery stores, you still have to travel far. But from my perspective, it's not really a food desert because they still do have access to fishing just outside their door. They still have access to hunting, game, and other wildlife nearby. So coming from learning from other tribal students and other tribal communities about food security and food desert conditions is kind of my approach to while I work on my PhD work. And then just incorporating it into our modern lifestyle. As we know, Native Americans or American Indians have the highest rates of like diabetes and other chronic diseases, but it's not our fault. Through history, we were introduced to this modern diet, which is very unhealthy. So how do we go back and live our ancestral diets again? And tribal food sovereignty isn't new. Like I said, there's a lot of achievements happening, a lot of programs happening. A lot of them are revitalizing their own Indigenous languages and their cultures, incorporating their elders, incorporating their youth within their food system. They do have a lot of motivation to protecting their land for future generations, incorporating and relearning traditional foods and plants and seed savings. And I can see that happening not just in the community, but like in academia, there are workshops to help those areas. And I think the knowledge has to be from academia to tribal communities, but also tribal communities to academia. And just being a Native scholar, a lot of Native people are succeeding in higher educations. Like it's definitely hard and challenging, but I was just thinking about this the other day, like Native people, Native scholars are meant to be graduate students. Like once we finish our 4-year degrees and we're on the baseline of knowledge as other people in academia, like that's where we can really focus our beliefs as Indigenous people, but in academia. And I worked a lot on this one sentence, just trying to figure out my approach. Like I said, tribal food sovereignty is constantly evolving. And my approach will be looking at natural and technical resources such as wildlife and agricultural land to the contemporary lifestyles of the tribal communities and efforts to preserve Indigenous culture, food, language, and ecological knowledge. So not just using my ag background, but incorporating all the resources that different tribes have and making it work for their modern lifestyle. And one of the biggest pushes I have a picture here of my family. We grew up farming a little bit. So my perspective is not only making it relatable and relevant to people in academia, but also to the res kids, to the kids growing up on the reservations, and making it possible for them to also work forward in tribal food sovereignty. So again, just by acknowledgements of all the school and campuses I have been fortunate to be a part of, scholarship and funding, the tremendous support of my family, friends, and communities along the way. And thank you. So here's my contact information.
Thank you, Nicole. And I'm trying to come back here. Okay, great. Wonderful presentation. I know if you have a chance there. Okay, excellent. Makani went ahead and answered Carrie's question in the chat. And we have time for maybe for like 1 or 2 questions. I'm going to go over a little bit more just because we got started a little bit late. But if anybody has any questions, you're welcome to unmute yourself and post a question to our 2 speakers.
This is Ken. I really enjoyed both talks. I had a question for Makani, and this is just a basic question. What is the role of moths in the overall ecosystem, the connected ecosystem? You know, I think of bees and pollinators and birds, but it could just be my lack of knowledge. But if you could just kind of give me, what is the role?
Yeah, no, I was chuckling a little bit because my advisor, Riley Nelson, would always answer those questions with to make more moths. That's the purpose, is to, to make more moths. But no, they're good for, and they have interactions for, they do get eaten by bats, even though they have those anti-predator defenses and other predators. So they are a food source for a lot of different things. And a lot of them do act as, again, pollinators. Like you mentioned, the bees, a lot of moths are pollinators as well. So they do have functions like that within the ecosystems. So.
Okay, we got one more question. If you want to go ahead and pose the question, and that way will take us a little bit over to 8:00. I'm going to end the call about 8:05. Okay, Carrie.
To Nicole, I wondered if there's a particular kind of agriculture that you're going to focus on, and whether you might carry over your cover crop kind of knowledge that you developed as a master's student.
Yeah, it's kind of hard. So my brain is like all over the place on what I can do and how much I want to learn as a PhD student. Definitely right now I'm working on networking with the Blackfeet tribe here in Montana. So in the beginning, I wanted to work back in the Southwest because that's where I'm from. And that's where I see myself in the future settling down. So I definitely wanted to pick a community to where I can work long-term. But I also think it's important for me to learn how to work with a new tribal community and learning their own ecological or indigenous practices. So I think that's going to be my focus, is learning other indigenous farming practices or collecting herbs, you know, not just looking at agriculture, but looking at their natural resources and their wildlife, and creating, I don't know, just having that experience for tribes to collect data on their own, relevant to their food system, and kind of applying that to being successful long-term for them, depending on their funding or how their tribe works. And I think that's what's gonna kind of help me here, is building the social aspect. So getting interviews as long as getting the biophysical science aspect. So I'm really interested in just learning more indigenous practices and kind of tailoring that to help the tribal communities in academia.
Great, thanks.
Okay, excellent. Well, thank you so much for your both your talks and for your questions. I do appreciate everybody joining us for this late hour here in the Eastern time zone, where it's just a little bit after 8 o'clock. And we do plan to have additional presentations as we progress through the spring semester. So thank you all again. I'm going to go ahead and stop recording just to close out this, just to say a formal goodbye and thank you all for joining us. And